All right, it's 12:01. Here we are once again. Uh, so we're here today for Brandon Fair's thesis project, just kind of the second half of what we saw yesterday with Catherine's. For the whole time, I've always viewed these projects as kind of twins, right? We're kind of answering very similar questions, but just in different ways. So I see all this is complementary. So thank you for being there yesterday, and thank you for being here today to see kind of the second part. We can see how the story unfolds, continues. Um, yeah, like I said yesterday, this has been a really cool project to see happen. It's really great to pair these eDNA and electrofishing surveys to get some methods work, but also understand some really interesting community dynamics along the way in a very, but I don't want to say peculiar system, but a system that's just quite different than what many of us kind of learn in these well-behaved dendritic upland kind of streams. Yeah, so I'm really excited about it. It's been a great partnership. Zenithia and Bill Sutton in the Forest Service team. So I guess a little bit about Brandon. It was August of 2012 when we first met. Yeah. I was just a year into my PhD at Virginia Tech, and I was suddenly informed that I would be teaching fisheries techniques about a week before class started. That was exciting. Um, but it was great. And Brandon was in the class. And it was great. It was like my first proper teaching experience. So I got to know the students a lot better than one might on just kind of a, ordinary teaching schedule. And, Brandon was one of them. And I was always struck by this guy who was like suspiciously optimistic. I'm like, I don't know. If somebody like gave me that much criticism on like an assignment or something like that, it'd be tough. Uh, volunteered on a lot of my work. I was doing this big experiment, so we were out in the field a lot, and it was just really cool. And I remember, I don't know if you remember, but the it was graduation in like May of 2013 or 14, 13.
13.
And I was walking home from campus, and there was this like I passed like this grill out, saw Brandon over there, and came over and we talked on the side of the street. And where are you going? Oh, well, I'm graduating. I'm going on a mission trip in Honduras. I'm like, well, that'll be the last I see of Brandon Fair. And then fast forward a little bit. What it's like 2021. Brandon's a crew leader on the U.S. Forest Service CAT team. I didn't know this. And CAT had come and help us with some work on our Bartram's Bass project. And I was seeing some pictures from that. And I'm like, that looks like Brandon Fair. And sure enough, the guy looks exactly the same, unmistakable. Like, oh, that's cool. He's kind of bubbled up back into this world. Fast forward 3 or 4 more years. Now we have this project in collaboration with Zenithia and Bill, and suddenly found ourselves in a situation needing a grad student very quickly. And all of a sudden, there became whispers from our friends in the Forest Service, various places. Hey, there was this, I got the perfect person. And usually, I like to do like all our own recruiting from scratch. I'm like, well, who is this guy? Now, coming from several different folks. Oh, it's Brandon Fair. You ever heard of this guy? I'm like, oh, I've heard of him. And so we talked, and it was just like, as I've said before, we couldn't have this project without Catherine. We couldn't have had Jacob's project without Jacob. This project literally wouldn't have happened without Brandon Fair swooping in and bringing the level of expertise and experience and maturity and just that indomitable optimism that I was skeptical of as a PhD student became an important glue that got this whole thing together. So it's really great to see what y'all have put together in no less than 4 semesters. On top of that, and so here we are. I've talked way too much about our history, and we need to talk about your work. So have at it.
All right. Sounds good. So, thank you, Brandon. I'd like to thank my committee, Zenithia and Troy, as well, for being here today, and everybody else, everybody on Zoom. Thank you for being here to support me and my work throughout my master's thesis. So, this presentation here is just the culmination of my thesis work in the coastal plain of South Carolina, exploring its unique fish communities. But first, a huge amount of field work went into this project. I couldn't have done it without my fellow graduate, Catherine Masatani. Thank you for all your encouragement on the way, introducing me to Spam Masubi. If you haven't had this delectable treat, I encourage you to look it up and try it. It's pretty amazing. And your positive attitude and hard work ethic is inspiring and it will get you far in the field of fisheries. Also, a big shout out to our technician, Adam Daniels, for sticking it out with us through crippling heat, venomous snakes, and a consistent plague of mosquitoes. And I mean consistent plague of mosquitoes. So we really appreciate all your hard work on this project and entering all of the data for this research as well. I'm extremely blessed to have my wife, Sarah, who's sitting here today for her just daily encouragement and support, as well as telling me whenever I needed a break because I'd been at the computer far too long and needed to go for a walk or clear my head or something. So I'm thankful for that. She moved with me from the mountains of Virginia all the way down to coastal South Carolina, and then 6 months later up here to Clemson, and that's no small feat. Also, my family for being ever present and just a phone call away. And I'd like to thank God for his guidance and faithfulness to me throughout this season of learning and growth in my life. Multiple students and faculty at Clemson for their input. A huge thanks to all the folks at the U.S. Forest Service for financial support for this project, as well as help in the field, multiple individuals at SCDNR for helping with species ID and site location. And last but not least, Bill Sutton and his Wildlife Ecology Lab at Tennessee State for processing all the environmental DNA samples for this project. And that is a 6-foot alligator in one of our sites. So I wanted to start off by telling you a little bit about a brief history of the coastal plain in South Carolina and its changes in land use over time. So historically, the Cooper and Santee River watersheds were deforested and developed for rice farming in the mid-18th century. Recent LIDAR data has been used to map around 95,000 hectares of rice fields in coastal South Carolina. A system of canals and dikes were created for irrigating these crops, which reshaped aquatic habitats in this area. In the 20th century, much of the cleared land was used for agriculture or reforested for timber production. Coastal streams are characterized by having warm temperatures, less distinguishable breaks between habitat types, lower gradients due to a lack of elevation, and generally are highly connected to their floodplains. They also have stream beds that are comprised of clay, silt, and sand, and fine and coarse organic matter accumulation releases tannins that create these unique blackwater systems. Streams also experience large amounts of variability with major droughts and floods, which is a core focus of this project, is really looking at that highly dynamic landscape. And this comes from storm events such as hurricanes, the floods do. So here's a quick overview of my thesis chapter. So for chapter 1, we focused on one species, the American eel, and did a gear comparison between environmental DNA and electrofishing, assessing detection efficiency. We also assessed important abiotic variables influencing occupancy and detection probabilities of eels. For my second chapter, we're going to zoom out a little bit to the community level to investigate the variables affecting within site intra-annual or within a year community variability. This was done both taxonomically and functionally. So taxonomically being with unique species and functionally with the unique traits or ecological roles of those species. So moving into chapter 1, the American eel. So human activities threaten aquatic organisms through habitat degradation and fragmentation, both laterally and longitudinally in streams and rivers. This fragmentation disconnects aquatic systems from their natural floodplains and creates barriers to migratory species that rely on connectivity, such as the American eel. And as climates and habitats change, it's important to regularly monitor fish species and study the factors that contribute to where species live and why they live there, which are core ecological questions. As the number of threatened and endangered species also grows, finding efficient and effective monitoring methods is crucial. So environmental DNA is an emerging tool that can be useful for monitoring fish species. In our study, this involves extracting genetic material from water that is shed or excreted from aquatic organisms. It requires minimal effort and is less intrusive than the more widely used electrofishing method. Also, research has shown that environmental DNA has usefulness in low species abundance situations as well as complex habitats and in accessible habitats. And also, it has applications for threatened and cryptic species. Using both environmental DNA and backpack electrofishing methods simultaneously can help us to understand factors that are influencing the distribution of this species. So the American eel, our study species that we compared these methods and detecting it, is one of a small list of catadromous species, which means that they spend most of their lives in freshwater habitats before migrating out to the Atlantic Ocean and the Sargasso Sea to spawn. This is the opposite life cycle as a salmon, an anadromous species. So I have personally helped on a long-term eel mark recapture study in the mountains of Virginia on the Tye River in a headwater brook trout stream where 15-plus-year-old eels were encountered often in similar locations to where they were marked originally. Because of this unique and highly migratory life history, American eels need effective monitoring. This map from Kwok et al. 2019 shows the American eel's native distribution from northern South America all the way up to Greenland. And it illustrates that they can be found across a wide range of habitat types. Barriers such as dams and road stream crossings have reduced the range of this species and contributed to population declines. A fun fact about American eels is they're extremely long-lived. I already gave you a figure of 15-plus-year-old eels, but they can live 20-plus years in freshwater habitats. And the oldest American eel that's been recorded in captivity was 88 years old. So understanding what affects the eel's distribution is important, especially in these dynamic landscapes like the South Carolina coastal plain that can experience major droughts that can lead to isolated pools and complete dewatering of stream channels, as well as widespread flooding from hurricane events that inundate the system, and urban development extending out from Charleston, South Carolina that channelizes, degrades, and disconnects these streams from their floodplains. Hurricane Debbie brought 22 inches of rain during our study, just as a side note about the flooding aspect. So our goal in the study was to examine gear-specific occupancy and detection probabilities of eels from site-level abiotic variables. We selected 45 sites and wadable streams that captured land cover types representative of the region. American eels were found at 34 of these sites, shown in white, the white triangles. Site locations in and around the Francis Marion National Forest ranged from heavily forested in the green to highly developed areas in the red. And watershed sizes ranged from 0.25 to 89 square kilometers. Each site was sampled 3 to 4 times over a 6-month period from June to December. And this period spans both the wet hurricane season as well as the driest month of the year, which is November. So for each site visit, we started by filtering 3 separate 1-liter water samples using the Smith Root eDNA backpack pump in the main current of the channel at the downstream end of the reach. Protocols such as not disturbing the stream, use of sterile gloves, and single-use filters help to minimize contamination risk. Next, we conducted a 2-pass electrofishing survey, followed by habitat and water quality data. Reach lengths usually were 100 meters with a couple around 170 meters, and our stream widths ranged from 0.9 to 7 meters across. So habitat data was collected at 10 equidistant transects throughout each of the reaches, flagged alongside the bank. So getting into electrofishing results for American eel, we captured 350 eels over the study period with electrofishing, with 228 of those being in our urban sites. Higher eels were captured across urban sites and across visits versus forested sites. It's important to note here that urban site locations retained water throughout the entire sample period. We never saw it go dry. For the number of detections at sites by visit between eDNA and electrofishing, eDNA had higher detections across all visits for the American eel. On visit 2, environmental DNA detected eels at 8 sites where electrofishing did not. This was following Hurricane Debby, which caused widespread flooding in the system. So taking an even closer look at this, looking at this contingency plot, which just shows the number of visits associated with each combination of detection or non-detection. I'd like you to notice that 28 visits had positive detections of eels with eDNA, where electrofishing did not detect them. There were also 3 occasions where environmental DNA did not detect eels despite finding them with electrofishing. So we used multi-gear occupancy modeling for this analysis, so I'd like to just very briefly go over that. So occupancy modeling estimates the probability that a species is present at a site while also accounting for imperfect detection. So this is broken into 2 aspects: occupancy probability, or what are the chances that a species is present regardless of if it is detected, and detection probability, which is the chance of detecting a species during a sample given that it is present. So this side-by-side multi-gear analysis was performed using the SP occupancy package in R to select ecologically meaningful site-level abiotic variables. We tested for collinearity using a correlation plot. And then we reduced the dimensionality of our water quality variables by running a principal component analysis and used the first axis for modeling. So for detection covariates that we selected from this process were temperature, because fish species can behave differently as temperatures fluctuate. Turbidity, which is a well-known variable affecting detection of fish species. And volume, because higher volumes at a site or fluctuations in volume can alter capture efficiency of fish species. Then for occupancy covariates, we selected percent forest as a gradient of land use, and also distance to ocean as a dispersal metric, since eels are catadromous and migrate from saltwater habitats into freshwater habitats. And then minimum volume to capture stream intermittency. We wanted to get at whether streams went completely dry during our sample period or they didn't with this. And then our water quality PC, which represents the dominant environmental gradient. So models were compared and averaged, and then top models were ranked by WAIC, which is an analog or the same as AIC in this framework. And then we took all of these models with a delta WAIC of 2 and then averaged those. So for occupancy covariates, we found that minimum volume and our water quality PC axis had significant effect sizes for both gears. And they also lined up directionally with one another on either side of 0. For detection variables, all had significant effects for both gears, and they also lined up directionally with one another. So I then created relationship plots to look at each method side by side for these significant variables. For minimum volume, occupancy probability increased for both gears with increasing stream size. But occupancy remains high across the range of minimum volumes. I'd like you to look at the y-axis. We're only moving from around 0.83 to about 0.95 with this increase. So very small increase. And this trend suggests that eels are broadly distributed in our system, and smaller, more variable streams only slightly influence their occupancy. Increased volumes in streams has been shown to provide flow refuge and hydrologic connectivity for eels. To understand our water quality result, the x-axis goes from sites with warmer temperatures, higher pH, and higher conductivity associated with our urban site locations to cooler streams with lower pH, lower conductivity associated with our forested sites. So there's a small decrease in occupancy probability in cooler sites. But again, the change is small, only about 0.1 or even less for environmental DNA. Um, and this suggests that eels they can tolerate many different water quality conditions. And we found them at high occupancy in all of our sites. So then for stream volume, there was a positive relationship between volume and detection probability. We had similar detection estimates as seen for both gears at smaller stream volumes, but then separation occurs at higher volumes. So there's a higher probability for electrofishing at higher volumes, and this is likely due to backpack electrofishing scaling up with stream volume in our wadable streams, but environmental DNA collection remained constant. We were still only collecting 3 1-liter water samples, no matter how much volume was in the stream. And also this lower amount of detection probability for eDNA could be because of challenges of transportation of genetic material or dilution in streams with higher volumes. And then for turbidity, there was a negative relationship between turbidity and detection probability, as expected. Similarities are seen again between gears until estimates drop dramatically for electrofishing at higher turbidity. Backpack electrofishing relies on visibility, so this extreme decline makes sense. eDNA is affected to a lesser degree, but this reduction, sorry, need to reveal that. But this reduction is possibly due to known complications with turbid water, clogging filters, or inhibiting qPCR amplification in the lab. So, and then finally looking at water temperature had a similar positive relationship with detection probability for both gears. There was a lot of overlap here as well. And again, looking at the y-axis, very small increase again in detection probability. So this is possibly due to higher water temperatures, increasing metabolic rates, swimming activity, and the shedding of genetic material that's been observed in other studies as well. So what are the implications from all this? Environmental DNA had higher detections overall for the American eel. If managers are simply looking for detecting eels in a system or to start an early monitoring program, then using environmental DNA would be a great tool for this. But ultimately, both methods came to similar ecological conclusions on factors that affect eel occupancy and detection in coastal streams. And taking this into account, managers should look at management goals and consider trade-offs between methods for monitoring design, such as available personnel, financial resource availability, and also time constraints. Eels were broadly distributed in our area below the Panopolis hydroelectric dam. And this area may represent what eel distributions could look like given full access to river networks. Maintaining connectivity and river corridors is important. So moving on to chapter 2. This is 37 of the 40 species that we found in the coastal plain, so a pretty diverse community. My favorite fish, if anybody was wondering, is the mud sunfish, actually, in the almost the bottom right, the large sunfish species down there. So my second chapter focused more broadly on fish communities and what influences their stability or variability within a year, so intra-annually. So a good portion of what we know about streamfish community structure comes from single season samples that assume variability within seasons will not overwhelm the overall patterns that we observe. This single snapshot in time is often used to draw conclusions about factors that shape these communities. But as I mentioned, the South Carolina coastal plain is highly variable with major droughts and floods from storm events and altered habitat from urban development. So community stability within a season may not always be the case. So our goal in this study was to first understand the factors of community stability within sites across visits over time, and then to test the effects of habitat variables, community richness, and dispersal on stability. There's really no good or bad guy with stability here. It just means that some communities are more variable than others. So we used a subset of 34 sites from our overall 45 sites for this chapter study analysis. Each site was sampled again 3 to 4 times over a 6-month period from June to December that spanned the hurricane season as well as the driest month of the year, November. So we used the same sites, the same field methods, minus eDNA, and the same data. So a 2-pass electrofishing survey was done, followed by water quality and habitat data. So looking at the fish community structure and what we captured with electrofishing, we had 16,400 individual fish that were caught with 40 total species represented. This graph shows the top 30 with over 6,000 mosquito fish. And we also caught some South Carolina state listed species, the American eel, the Everglades pygmy sunfish, the iron color shiner, and the white catfish just outside of this graph. And actually, the Everglades pygmy sunfish, there weren't many records in South Carolina, so it was really cool to see this species. And there's actually a student here right now, Adam, that will be looking at Everglades pygmy as well as other rare and threatened species in this area. So then looking at species richness within sites over time, we saw as many as 0 species to as many as 16 species in some communities. This gives us an idea of the very dynamic nature of coastal plain community structure. So in order to interpret the results of this chapter, we first need to understand 2 different fish diversity metrics. The first being replacement or turnover. So this can be seen as a compositional change in a community, either taxonomically through unique species turnover, replacement, or turnover of functional roles in an ecosystem or traits. So for example, in terms of our study, if in visit 1 we captured 5 species, but in visit 2 we observed species F and G instead of A and E, then this would be a taxonomic species turnover. But taxonomic turnover is not the same as functional turnover. Species E might share similar ecological roles or traits to species G, but A and F do not. And then if in visit 3 we captured 5 completely new species, this would be a complete taxonomic turnover. So then looking at richness difference as the second diversity metric, this can be seen as gains and losses of species within a site over time in our study. So both taxonomically with unique species or with traits functionally, but it doesn't tell us which species were gained or lost, just that which species or traits were gained or lost, just that they were gained or lost. So if in visit 1, we had 6 species, but on visit 2, we only had 2 species, this would be a taxonomic loss or richness difference of 4 species. But if in visit 3 we had 8 species, this would be a gain of 6 species taxonomically. But again, functional and taxonomic are not the same, and 4 species in visit 3 could share similar traits or ecological roles to one another. So we used the correlation plot again, just like the first chapter, to select variables and then that represented natural change, land use, and dispersal potential. We then used the water quality PCA in order to reduce the dimensionality of all of our collinear water quality variables. And the variables that we selected for this were percent forest, so representing that land use gradient again, or anthropogenic disturbance, substrate diversity, just representing habitat diversity and how different substrates can structure fish communities in different ways. Our water quality PC as the dominant environmental gradient, minimum volume to represent stream intermittency in our sites, and then CV volume to represent variability in volume. Max flow was selected to represent disturbance magnitude, and CV flow for that variation in flow. And then max species richness was selected. So this was actually chosen because beta diversity components like replacement and richness difference can be sensitive to changes, can be sensitive to richness difference changes, both taxonomically and functionally within sites across visits. So for example, if you had a poor richness community with 3 species and you added 1 species, this would be a greater proportion than if, say, you added 1 species to a more rich community that, say, had like 10 species. So, and then distance to river was chosen as a dispersal metric, and we use this as an interaction term in our models with every variable to see how stream network position affected streamfish community structure. So after this, we calculated 2 different components of beta diversity that I talked about, turnover and richness difference, both taxonomically and functionally within sites across visits, and this yielded us 4 different ways of assessing intra-annual community stability. Then we built a global model of selected variables and used an all subsets approach in order to select top multiple linear regression models by AIC. Models with a delta AIC of less than 2 were then averaged so that we could interpret significant effect sizes and variables from this. So our average top models revealed that the interaction coefficient, distance to main stem rivers, and maximum flow was the most significant. Distance to rivers here was defined as a greater than or equal to Strehler order 5 river, so a large main stem river. So to orient you to these results graphs, as we move up on the y-axis, communities become less stable or more variable. We broke our distance to rivers out by standard deviation, with a +1 standard deviation meaning high distance to rivers and a -1 meaning low distance to rivers. So we did this for interpretation. So first thing I'd like you to notice here is that when you are far away from rivers, more flow causes or reduces variability and turnover in communities. So if you have a good stream with sufficient flow in this context, then site species movement and swapping is reduced. But the opposite occurs whenever you are close to rivers, and more flow actually creates more variability in these sites. So when it's a nice stream with good flow in this context, dispersal potential increases. So then looking at significant variables in taxonomic average richness difference, distance to rivers and both max species richness and percent forested land cover were significant. So looking at max species richness first, when you are far from rivers, richer communities are more variable because species losses cannot be compensated for from a colonist pool from main stem rivers. But as you move closer to rivers, this distance to river acts with a stabilizing effect, and there's little relationship between max species richness and average richness difference because species losses can be compensated for from movement from a colonist pool. So for percent forested land cover now, what I'd like you to notice here is that when you are far from rivers, more forested land cover was associated with less community variability because species in these remote locations are adapted to local conditions in these headwater isolated sites. But as you move closer to rivers, higher forested land cover actually increases variability in communities. So again, if you have a good stream with good flow and good forest land cover, it generally can support more species movement in and out of sites close to rivers. So then moving to the functional dimension, we looked at functional average replacement, and we found that percent forested land cover had a significant effect size. So breaking this down, locations with less forest land cover have less functional replacement occurring in the community, possibly because new species that return after disturbance events could share similar ecological roles or traits. But in more forested locations, there's an increase in functional replacement, which suggests that these sites have a higher chance that functionally distinct species will replace one another. So then finally, we looked at functional average richness difference. Significant main effects from this analysis were distance to rivers and the coefficient of variation of volume or hydrologic variability. And then the interaction between distance to rivers and max species richness. So sites closer to rivers, looking at distance to rivers here, sites closer to rivers experience higher trait fluctuation between visits, potentially due to dispersal movement from a colonist pool. Got a lot of species moving around. But in contrast, sites farther from rivers had less trait fluctuation, suggesting less influence from dispersal and more specialization as species experience more habitat or local filtering at these sites far away from rivers. Oh, gosh, I missed a lot there. All right, so sites looking at coefficient of variation of volume, sites with low CV volume variation showed lower functional gains and losses, suggesting that species with suggesting that stable flow conditions in these environments may limit the gain or loss of species with unique traits between visits. But then sites that had higher CV volume or higher variability or intermittency had greater functional gains or losses between visits, suggesting a higher probability of distinct species and dynamic flow regimes. Then finally, we looked, or this interaction effect between max species richness and functional richness difference. What I'd like you to notice here is that when you are far away from rivers, richer communities allow for more subbing in and out of species with unique traits in between visits. This observation is similar to one seen with taxonomic richness difference, if you can remember that plot, where you not only lose unique species, but their unique traits further from rivers. But when you are close to rivers, richer communities mean less swapping in and out of unique traits between visits because those species simply just remain at these sites between visits. So I beat you over the head with a lot of graphs. So what does this all mean? Variable landscapes experience intra-annual fluctuations in community stability, which can be very important to understand for managing these species in these environments, because a single sample per year will not capture this variability. The taxonomic dimension was influenced by the interaction of environmental variables and dispersal. So maintaining hydrologic connectivity, especially to mainstem rivers, is important. Our headwater streams in this area are vulnerable to species losses as well as variability. Also, the protection of forested land cover is critical in sites far from rivers where it can have a stabilizing effect on communities, and close to rivers as well, where it can contribute to resilience and dynamic communities as these species move around in and out of these sites close to rivers. Functionally, for our functional dimension, it was influenced more by habitat filtering on a local scale. And more variable streams supported higher functional diversity and specialization. So these fluctuations that we experienced in the South Carolina coastal plain with your drying and your wetting of stream channels is actually very important to coastal communities, especially in these isolated forested habitats where species are very specialized and very adapted to their environment. And then finally, the taxonomic and functional dimensions offered us valuable insight for streamfish community structure. And they gave us different insights into this community structure. Again, with the taxonomic side of things, environmental variables interacting with dispersal processes, while on the functional level was more, the main effect there was with local habitat filtering. So these are the takeaways from my talk today. And with that, I will take questions. Thank you. So if you have any questions on the chat, you can just type it in. I'll try to get to those. All right, Colby.
Just wondering what your overall recommendations would be, like, given the turnover that you see throughout the year for community monitoring in that area, like what would be your suggestions to agency biologists interested in knowing what's out there, I guess,
Yeah. Yeah, no, definitely makes sense. To plan multiple visits throughout the year would definitely be a recommendation of mine. And to be in tune, I guess, with fluctuating water levels in your system as well. So when water returns to some areas that maybe have been in drought conditions for a while, potentially being ready to go in and sample some of those sites just to see what fish move into those sites after drought events. So yeah, definitely multiple times a year.
Great talk. I was just wondering, when you're going to a site and you take eDNA versus electrofishing, what is the time difference between taking eDNA sample, processing it, and then shocking a site and then processing this thing?
So quite significantly different. And it varied a lot because our streams, as I mentioned, range from like 0.9 to sometimes 7 meters across. But in general, the environmental DNA was just consistent unless we ran into any sort of problems with maybe flow rates of filtering being a lot lower. It might take a little bit longer, but essentially no more typically than 5 minutes to get through, 5 to 10 minutes for environmental DNA, and then you're out. And that's for 3 separate samples. So if all is going well, but with electrofishing, sometimes it could take as many as 2 to 3 hours if we were in a larger stream, right? So we had one particular stream. I was actually the one shocking the 2 passes, and we did spend about 3 hours just shocking, let alone working through the fish. So very much a half day site. Yes.
You seem very calm up there, but not even course, so that's
Whether or not I feel that way.
in your first chapter where you're talking about water quality, it shows that there was less observation by shocking, but you still retained the DNA observations at low water quality. Is water quality associated with further downstream sites? You assume that the reason you're seeing the eDNA persist there because at upstream better quality sites, the eDNA is flowing down. I'm not sure if
I remember correctly. Actually, the trends were on top of each other. The confidence intervals lined up. There was a decrease moving towards our more forested sites, but very slight. It was around 0.1 or less occupancy probability drop between the gears. So there really wasn't much variance with that, and they came to the same, pretty much the same conclusions, both methods. Yeah.
So great talk.
Thank you.
Really good. The going to the detection probability part of it. We saw a decrease. You saw a decrease with eDNA detection as water volume got larger, correct? that's something I had never thought about before. I've taken a lot of EDNA, but I've never really thought about the influence of like flow. What would you recommend more, more sampling of EDNA in larger rivers? Compared to
Yes, actually, and I thought about this a lot throughout our project. So our samples we took 3 different samples, and typically our streams were not super large. We did have a couple sites that had that 170-meter reach, and those were a little bit wider, but typically they were small enough where we could get our samples in the main current and feel pretty confident that we were sampling that stream well. But in larger streams, I did think about whether spreading out our samples across the current, because you have different transport of that genetic material in the system, depending on where you're at, where the eddies are, like how the currents are moving in that river, as well as a lot of dilution as well from higher volumes potentially, especially if you have a river that increases in volume, but your abundance of that fish species stays the same. There's going to be a lot more dilution of that genetic material because you just have a lot more water for it to be mixed into. But yeah, I would say that larger streams, maybe looking into multiple locations along a transect potentially, could be something to look into. And I have seen that and heard that in some other talks as well, of people using that in larger rivers or streams.
So I have another EDNA question.
Okay.
But first I want to make sure I got the water quality PC one right. So you had a decline in occupancy of both EDNA and electrofishing.
Yes.
At higher water quality. And that included phosphor, but it also included pH, and it was a lower pH, right?
Yeah, more acidic.
So knowing from the literature, EDNA degrades a lot more rapidly in lower pHs, more acidic systems.
Yes.
Do you feel like that could be contributing to your lower occupancy at all?
So, we didn't believe so, mainly because of how slow that the water moves in these lower gradient streams. So anything that we pick up, really, it probably hasn't flowed very far down the river system from wherever that species is that got detected, right? Because as you increase flow rates, you're going to increase the distance that genetic material is going to travel downstream. Otherwise, because eDNA can be, the particles can be different sizes, so they can settle out actually onto the bottom at different rates. But no, we didn't believe that it was like a huge factor. Go ahead, Brandon.
Did occupancy for backpack shocking and EPA differ from one another along that water quality grade?
No, they did not. No. Yeah.
For your occupancy, great talk, by the way. I think it increased with water volume, correct?
Yes.
Detection
Yeah, for both, but there was separation that occurred more so with backpack electrofishing, actually.
Okay, and how did you calculate that water volume?
So, so that water volume was calculated. We took widths at 10 different transects, and then we took 3 depth measurements along with flow along each transect, and then I calculated water volume actually for each point. So that would be like 30 points, right? And then averaged all of those to get like one water volume for that stream.
Do you think those ideals are select for section or water volume?
So potentially so, and actually there were less eel detections or occurrence. So if you remember back to the original map, we caught eels at 34 sites. The other sites, many of those are very upper headwater streams that experienced a lot of intermittency, maybe even multiple droughts or complete dewaterings throughout the year. So, yeah, which that did show up. Minimum volume did show up in our occupancy covariance. As those minimum volumes get towards like zero, you have a drop off in occupancy probability of eels. So kind of getting at that a little bit. All right, anything else? Yeah.
I have one more.
All right.
Why do you fish people keep thinking that running into cottonmouths in the field is a bad thing?
I actually love cottonmouths. I think they're really cool. My wife does not agree at all. But about Adam and the Probe. Oh yeah, we were shocking one site, and he comes around like this little corner, right? And just straight into a cottonmouth, actually like hits it with the probe, right? And that thing like freaks out, and he freaks out, and we're back there with like the fish buckets trying not to dump the fish. And so yeah, but regularly ran into them. Thankfully I've never been afraid of snakes, which Brandon was talking about that I was kind of fit for this project. Well, I've worked in the Francis Marion before. I knew what I was getting into, and thankfully I'm not too afraid of them. So it helped me out. So, all right, let me check the chat because we got some stuff here. All right. All right, so all right, so did you find any invasive species in your studies that may be harming the American eels, swamp eels, snakeheads? This is from my Uncle Roger. No, no, we did not find any invasives such as the swamp eel or snakeheads, thankfully. So nothing like that. But actually, my co-graduate Catherine did find a green sunfish, but not really something that is extremely affecting the American eel. So cottonmouths and mud sunfish rule from Kevin Kubach. Let's see, did you notice a trend in state species of concern and community structure? This is from Keith Whalen. May have to clarify that, Keith. You talking about the state species that I mentioned as far as the Everglades pygmy, the iron color shiner, those species? Yes. Okay. No, we don't have any results from that yet. I'm trying to remember if we actually were doing qPCR assays. Zenithia or Brandon, did we add those to the list at all? Okay, so we're still waiting on metagenomic data from Bill Sutton, and we'll be able to look at community structure specifically for those species, Keith. All right, and then, okay, I got one here from Colin Kraus. Let's see. Could you please re-explain the concept of near river versus far from rivers? I think I missed that explanation. Okay, so we broke our distance to main stem rivers, which that's being defined as a Strehler order greater than or equal to 5 river. We broke that out by standard deviation from the mean, right? So calculating along, using a DEM, we created a stream network, and we calculated the distance from each site to the main stem greater than or equal to 5 river. So we took the mean of that, and we broke our distance to river out by standard deviation, with a +1 meaning high distance to rivers, and a -1 being low distance to rivers. Does that make sense? Did I explain that okay? Okay, cool. All righty. Well, that's all I see here. If there's nothing else. Yeah, okay.
I've shocked up American eels probably maybe twice. My time electrofishing. I was in the 2 times we shocked them, they went pretty ballistic and shot away. I'm like most fish that do a little twirl or something that go up right. I was wondering if that is how they behave, or I was just kind of curious how they respond to electricity. when you're shocking, if they're well behaved or not so much.
Well, I'm making this comparison, but
Yeah.
but you would have to know how snakes get shocked. But so a lot of times they will roll over, and then you'll see their white bellies, and they'll kind of be in like an S shape or something because they're getting hit with that electric
Yeah,
current,
yeah.
right? A lot of times you'll see them like that. But you're right. They can be very elusive sometimes in getting through your electric field and current. And that's one of the difficulties of sampling eels in the coastal plain, because when you're dealing with black water systems, you can't really see the bottom very well. And so you can't tell whether or not an eel's getting by you or not with electrofishing. And so that's why we wanted to compare environmental DNA with that as well, because you're just sampling the environment, right? Like the genetic material in the water versus relying on that visibility to find that species. So, and we didn't know how many eels that we would catch exactly. I mean, they can be fairly cryptic and elusive in nature, but we were pretty good at catching eels, as you could see from our occupancy probability plots. And so they're not rare or threatened enough in the coastal plain where we weren't able to efficiently catch them with both methods, even with visibility aspects for backpack electrofishing. So, okay. All right. Thank you very much, everyone. Thank you, everyone. I'm going to end the meeting right now.